Can everyone hear me well? I'm hearing a bit of an echo, but okay. Hi everyone. I'm very happy to be given the opportunity to speak at this conference. I'd like to thank the organizers for that. So we can go on. So, as most of you know, most phenotypic variation in complex traits cannot be explained by studying the effects of single genetic loci alone. And one of the endeavors essentially is to go into interactive effects, epistasis, hopefully to unveil the missing heritability. So we're looking at a model that's a departure from additive effects of SNPs. For now, we're just interested in pairwise SNP interactions. And the standard way, how people are modeling this, is essentially, a very straightforward phenotype equal to some bias or intercept plus the first coefficient times the first SNP, second SNP, and then this is where it becomes interesting. We're looking at a multiplicative effect where the SNP A is multiplied by SNP B, and this is what we dub as interaction for now. And very simplistically, just if you were to imagine the SNP by SNP matrix, we're only interested in the upper triangular matrix for non-unique SNP combinations. And this is of an order of n to the power of 2, if n is the number of SNPs you have. So in human studies, we're looking at in the range of nearly 10 to the 7th SNPs over thousands of individuals. That leads to 10 to the 14 pairs of SNPs, and that is very computationally expensive. So one of the there's two main classes of approach on how to tackle this problem. Essentially, one that would limit the look at a subset of this larger combinations, and that's dubbed under space pruning. So in space pruning can be further broken down into filtering approach by either known biological pathways or by filtering by statistically significant main effects. So referring to the previous model, we're looking at main effects like SNP A and SNP B being significant, at which point it passes a threshold and goes to another subset where they do the interactions amongst themselves. The other approach is index structure approach, at which point they do branch and bound, and they do efficient updates of contingency tables. These are published by Zhang et al. In 2010, but it's still very limited from our regards because it's limited to discrete values, discrete phenotypes, discrete genotypes. We're interested in dealing with the exhaustive search, so scan for all possible combinations. And at this point, if you're to do it across all genome, it poses as a computational challenge because it would take nearly or greater than a year to compute on a single core CPU, and as a statistical challenge because once you have all these results, how do you interpret them? You have this Bonferroni multiple correction factor, but that's overly conservative because many of your combinations involve the same SNP. So we're favored maybe something more of a permutation-based testing, but that overall, that at the end of the day leads to another computational challenge. So our problem statement at this point is to implement a brute force, exhaustive, two-locus interaction multiplicative effect search algorithm. And it has to be time and cost efficient, and has to be able to accommodate for a quantitative phenotype and genotype. Quantitative, I mean real values. And has to be an in-house solution due to the confidentiality nature of our work at the Institute. So along came graphics processing units, and traditionally you would imagine graphics processing unit, the main use of it is to render graphics to your monitor. Here I have a little quick example that we're all used to. And over the past couple of years, the gaming industry and the graphics design industry has really perfected this field. So what does this really have to do with us, right? So if you were to imagine all of these data points essentially all following a single instruction, then we can apply the same strategy to our search because every single interaction is independent of each other, and if we can somehow just put in one instruction or one function to it, then essentially we're doing the same thing. If we're to do the same function to the CPU-based computing, you see a very saccadic, very slow process. And CPU is really essentially not meant for this. So this is something that you can plug into your desktop computer through the PCI Express bus, and off you go. So instead of playing games, you do computation, or we're supposed to. So GPU computing can be thought of as composing several hundred very lightweight processing units. And contrary to popular belief, there's still the need, of course, of CPU. CPU is there to coordinate everything. It's meant to run the sequential part, and GPU handles the computationally intensive part in a single instruction, multiple data manner. So other benefits is the cost of CPU. Once you factor in the performance, CPU is much more expensive than GPU. This is of importance, especially these days with the economical status that we're in. Flops per euro, this is just a very, very generic snapshot of what I saw on the internet. It's roughly, we can boil it down to 7.5 gigaflops per euro for GPU, and CPU is only 0.25 gigaflops per euro. Other benefits include lower power consumption, so you don't have to worry about building something very specific for your cluster of computer. Lower heat dissipation, less space requirement. These are three GPUs that we have neatly tucked in a desktop machine. In terms of programming language, there's many of them. These are just a few of the most popular ones. And at the heart of GPU programming is that you want to keep all the threads, all these GPU threads simultaneously busy. And you also want to minimize memory access, because the memory latency to communicate between the host and the device is very, very sluggish. Here is just a brief illustration of what we're doing. So on the plane, you see the SNP by SNP matrix once more. And due to the memory limitation of the GPU, you can only load partitions of your SNPs into the GPU at a time. So once you load them in, it goes into what they call GPU blocks. The blocks in turn are composed of many GPU threads. The take-home message here is essentially there's this hierarchy, because once you go into all the GPU threads, you have to somewhat resolve back to what is related to which interaction at the end of the day. And there's a whole community on this, gpgpu.org, that you can read up on tutorials and optimization tools. So in short, the GPU provides impressive computational power, but it's most effective when you can break up the task into many sub-tasks that can be done in parallel. So since the significance of each of our SNP pair can be tested independently, this is really well suited for the GPU computing. Our implementation is done in R. We've modified this CUDA wrapper GPU tools. We also accommodate for population stratification by doing an on-the-fly meta-analysis. This is important because we cannot afford to keep all the results for all the study independently, because it would just blow up your storage. And this is something that we're very confident about. In terms of the actual equation that we're fitting the problem to, our first initial attempt was for binary phenotypes. So this is what we call epiblaster, and we're essentially looking at a difference of measure. This is the difference of measure between SNP1 and SNP A and SNP B, and the cases controlled separately. And then we moved on to quantitative phenotype, at which point we ran out of really creative names, and we called it epi-GPU-HSIC. It's going to be apparent why the name comes later. So at this point, you can see that the computation is very simple. It becomes just a summation of all the scaled and centered SNP A and SNP B with a phenotype to the power of 2. And this is very, very, very efficient for us. An overview of the two methods. This is just to illustrate that we essentially boil it down that these two methods are not that different. once we recognize that there are both instances of what's called Hilbert-Schmidt independence criterion. In short, Hilbert-Schmidt independence criterion is to do a squared correlation of two random variables in some feature space, and our two random variables in this case is the interaction term and the phenotype. So by playing around with the mapping of the phenotype, you can see that it's essentially the same thing, but one that's more applicable for binary phenotype, another one that's more applicable for quantitative phenotype. And for now, we're only limited ourselves to linear kernels, but in the future, to write it in this manner, we can think of many more creative ways of testing for statistical independence. So in terms of validation, we still pin ourselves against the standard, the golden standard, which is the p-values of the linear regression on the coefficient term. And in this case, as you can see, the x-axis is just the H6 score, and it's an R-square of nearly 1, or I think 96.7%. But these are just simulation data. And in terms of its performance, it scales linearly based on the number of SNP pairs we're doing. This is for also to note that this is for 10,000 subjects, and it scales really linearly. Okay, once again, I'm bringing in the cost factor. So for the 10,000 subjects, we're looking at roughly to calculate GPU HSIG, 70,000 interactions per second. And if you were to factor in the cost, we're looking at roughly 350 interactions per second per euro. And if we do the same thing on CPU, we can only achieve 800 interactions per second. And that's a factor of, if you work it out, you have a speed up factor of 90, but this GPU setup is actually 3 times more expensive than the single core CPU, so you have an overall cost benefit of 30. The next step that we did was to apply it to real data. And working at the Psychiatric Institute, we came across this psychiatric trait from major depression. The overall clinical objective, as most of these genetic studies involve, are to tailor some kind of treatment for the patients with major depression based on their genetic makeup. And the depression assessment that we used in this case was a Hamilton rating scale. So what is a Hamilton rating scale? It's a very subjective. Well, okay, I shouldn't put in too much of my opinions in this, but essentially it's a questionnaire that the clinician assess a patient based on 21 questions, based on their mood, feelings of guilt, and insomnia. And the data set that we have consists of 491 patients that are currently undergoing antidepressant treatments. And these antidepressant treatments, there's a whole slew of them. There's a various type, many different type of antidepressant treatments. And nearly 500,000 SNPs. And the phenotype that we took in this case was a week 2 percentage change compared to the baseline week 0. So we also pin it against the state-of-the-art method that's published by Schubach and all in 2010, dubbed fast Epistasis. This is an optimized multi-core CPU solution, and in this case we use 48 CPU cores, and these are the runtime. For our single GPU unit, it took 40 hours to run, whereas under 48 CPU, it took 57 hours to run, and this is nearly a factor. If you were to factor it down to a single core CPU, we're looking at something like 70 times faster. And here we're plotting the HSIG p-value versus the linear regression p-value obtained from fast epistasis. Not as clean as simulation, but as you can see, the area of interest, which are the high p-values in this case, they fit pretty well. We're pretty confident that we're not losing any significant pairs that are isolated by fast epistasis. So in terms of results, our findings, we found that there's interaction between the PBX and MEIS genes, and these genes had previously been noted as involved in brain development. So this further augments the hypothesis that there's interactions between these two genes. Our current status on this project is that we further implemented essentially linear regression on GPU, which in this case, again, very creatively, we call it epi-GPU length. is currently under review. And now it does a full linear regression on the GPU, and it's implemented in CUDA-C. We forgot about R for now because we're really looking for speed and ease of use for everyone, and has an additional speed of factor of 5. So if we're looking at, in this case, we're scanning through many numbers of subjects. The circles represent the GPU results, whereas the triangular ones represent the single core fast epistasis result. And the last plot is the standard Planck epistasis. And when we look at the speed of factor, epi-GPU compared to Planck is nearly hovering around 2,000 times faster. Compared to fast epistasis, we're looking at roughly, this is a log scale, so it's roughly, 150 times faster or so. 100. Okay, so in terms of conclusion, we've successfully developed an exhaustive epistasis search on consumer-grade GPU. I stress consumer-grade because we want this method to be accessible for everyone. You don't need to specialize. You don't need to buy a specialized type of GPU, and reduce the search time down to a single day for genome-wide search. We further extended the difference of coefficient method for binary phenotype to quantitative phenotypes by recognizing that they're both instances of HSIC. So future directions, we're looking at implementing gene-environment interactions, looking for data, because we now have the capability of investigating inputs of real value. We can really apply it to many different things, brain imaging, environmental factors, anything that we can think of that may have an interaction. Higher order interactions, so potentially rephrasing the question to handle higher order, third or fourth order interactions. And also to apply nonlinear kernels to extend the framework to quantitative phenotypes at the time series or that are with images. Here's just an example of something that we can apply. The time series, these are actually the Hamilton scores, and they're scored every week, weekly. It's a real mess. It looks like a real mess. There's a general decay of the signal, but yeah, we have to look into this a bit more. And here's an example on the left side. You can see these are just, here are just a 2-SNP, and we're plotting a violin plot on the different points. And this is a surface of what it fits to. And at this point, we don't have any points to support it. So theoretically, it shouldn't be a surface of this nature, and potentially more of a surface that looks like a Gaussian, bimodal Gaussian surface, something like this. And availability, the two softwares are freely available to download at these sites, and the third one is coming up too. And yeah, so these are my acknowledgement. I would like to thank Bertrand Müller-Musock, who is my advisor at the MPI of Psychiatry, Statistical Genetics Group, Bernhard Schultkopf and Carlsten Bogworth, who is in attendance here from the MPIs at Tübingen, and and everyone that we have collaborations with. Thank you.